Hi and welcome to the 1.1 introduction to the specification. If you want to read through it, I won't go through it, but you can pause and read if you would, and the rest of the spec is just there. Hopefully, I am going to summarise this section into key knowledge that you need to know that will help you to get the marks. So, let's start with cell theory. So, cells differ hugely in their structure and function, but they all share the same properties. Or they all share some properties, sorry. So, for example, cells have different structures and functions in different organisms. They have different organelles. However, there are 4 properties that every cell has to have in order to be a cell. So, they should all be surrounded by a membrane. The membrane will hold a cytoplasm where the chemical reactions take place. The cells have to be the smallest units of life, and they also have to be able to produce their own energy. You'll learn a bit more about energy production later on, and also chemical reactions in the cytoplasm. In science, there's always an exception to every rule, so there are exceptions to the cell theory as well, and there's a few that the IB want you to know about. So the first exception is skeletal muscle cells, and they're an exception to the rule because they're said to be multinucleate, which means that each cell has more than one nucleus. Or if you look at a diagram of muscle cells, you'll see that the cells kind of seem to run into one another, so they're not defined as individual units with one nucleus each. It just looks like they have many nuclei. Giant algae are much larger than other cells, so they're actually visible with the naked eye. And obviously, cells usually aren't visible with the naked eye. And aseptate hyphae are separated into septa by cross walls, which makes them different to other cells because they're not separated by a cell membrane or a cell wall. Okay, a couple of terms that we need to know. So unicellular is the clue's kind of in the name. Uni means one and cellular refers to cells. So that's an organism made up of one cell. Multicellular is an organism made up of many cells. Again, multi is many, cellular is cells, so many cells, multicellular. One concept that's quite hard to get your head around is the concept of emergent properties within organisms. So emergent properties refers to the fact that the whole has properties that individual parts do not. And a really nice example is the fact that heart cells have the ability to contract by themselves, but they can only actually pump blood around the body when assembled into a working heart. So the sum of the heart as a whole is more than the sum of its parts individually, because obviously the heart has functions that those cells don't have individually, and that's what we mean by emergent properties. You'll see that crop up in exam questions here and there. Okay, surface area to volume ratio. So we need to think about the size of these organisms that are made up of cells. So in small cells, molecules can diffuse in and out of the cell and reach all areas. So if you have a look here, if you have a really small cell, you've got the nucleus in the middle, you can see that it wouldn't take much effort for a molecule to get into that cell and straight into the nucleus and into the middle of it. In larger cells here, obviously, any molecule is going to need to get into the cell, and then it's going to take a little bit longer to make its way all the way through the cell to the center. The same is true for small and large organisms. So small organisms, things can diffuse, molecules can diffuse in and out quite happily. However, in large organisms, you need a transport system. So, for example, we have the blood in order to transport things around the body, because things can't diffuse from outside of your body right into the middle without being transported. So we need a mass transport system. So we therefore say that small cells have a large surface area to volume ratio. So this one here has quite a large surface area in comparison to its overall volume, whereas this has quite a small surface area in comparison to the actual volume of the cell or of the organism. So if you think about it, mice would have a small surface area to volume ratio in comparison, a large surface area to volume ratio in comparison to elephants, who have quite a small surface area to volume ratio because they have not so much skin in comparison to, relative to, the amount of cells that they have within the body. So it's all, surface area to volume ratio is all relative. Okay, let's have a think about microscopes and actually visualizing these cells, and how do we know about the structure of cells? Well, we know because we're able to look at them under microscopes. There's two different types of microscopes: a light microscope, which is what you'll be used to using in a school lab, and an electron microscope, which you might not be familiar with because you only find those in working labs and the university labs. So a light microscope uses light to visualize a specimen. The clue is in the name there. So light shines through the specimen and visualizes it in that way. In order to let the light pass through, the specimen has to be really thinly sliced, because obviously if it's not, the light won't get through and it won't visualize it. Your specimen is mounted on a microscope slide and might need to be stained or dyed in order for you to see it. So often you can't see the components of cells without staining them with specific stains. You need to know the different parts of the light microscope and how to use it. So there's a little diagram here, but obviously you'll do that in your school lab, and you'll learn how to use it and visualize cells. Okay, when you do this practical, you will be required to draw some diagrams of what you see. So you need to ensure that you use a sharp pencil so that the lines are really clear, and the lines have to be continuous. So you can see that this is a nice drawing, this not so much. So the lines aren't continuous. So you can see that there's a join here. We don't want that join. There's another join there. We don't want to see those joins when you're drawing diagrams from microscope slides. The line should be drawn freehand, not with a ruler, but when you label, they can be drawn with a ruler. So these label lines are straight lines drawn with a ruler, but the actual drawing is freehand. And don't put any arrowheads on your labels either, because that's scientifically incorrect. Okay, you can also calculate magnification. So this comes up in exam questions quite often, and it's usually worth 1 or 2 marks. Before we look at how to calculate magnification, you need to know that 1 millimeter on your ruler is equal to 1,000 micrometers. So if we split that 1 millimeter up into 1,000 tiny micrometers, you can split 1 millimeter up into 1,000 tiny micrometers. And then 1 micrometer, so 1,000th of a millimeter, can be further split into 1,000 nanometers. So just keep that in mind. You should always measure your diagrams with a ruler in millimeters. So don't measure in centimeters. If you do measure in centimeters, make sure you convert it to millimeters. And also make sure that when you calculate your units, your numbers are in the same units before you start the calculation. So here we've got the triangle for magnification. So magnification is M, which if you then cover that up, M equals image size over actual size. So I is the image size, and that's what you would measure with your ruler in millimeters. And A is the actual size, and that's generally given to you in an exam question. So that would usually be given to you in micrometers. The image size you would measure in millimeters, and then you would need to convert it into micrometers. So for example, if your image size was 56 millimeters, that's going to be 56,000 micrometers. We've got an example here. So if our mitochondria has an actual length of 12 micrometers, but when you draw it, it's 8.4 centimeters long, we can calculate the magnification as image over, they've called it specimen size, rather actual size. So you've got image size was 8.4 centimeters. So that's converted into 84,000 micrometers. And then you just divide it by the actual size, which was given up here, 12 micrometers. And then the answer would be that the diagram was magnified 7,000 times. It's worth practicing magnification questions. Okay, last bit of 1.1 is stem cells. So you probably will have heard of stem cells before, and you will have done a little bit about them in previous study. Stem cells are cells that can differentiate into a number of different types of cells. There's 3 different types of stem cells as well. So you can get embryonic stem cells, which are found in early embryos, which is embryos up to 5 days old, so like really fresh embryos. They're totipotent, which means that they can differentiate into any body cell. So embryonic stem cells are really special because they can differentiate into any type of body cell. You also find stem cells in the cord blood, so they're taken from the blood within the umbilical cord at birth. They can be frozen and used later in the baby's life for specific treatments, and they're said to be pluripotent because they can differentiate into all body cell types as well. Adult stem cells are found in some adult tissues, for example, in the bone marrow. However, they're difficult to obtain and they're multipotent, which means that they can differentiate into only a few different cell types. Now, the IB want you to know about two different diseases that can be treated with stem cells or with stem cell therapies. The two diseases that you need to know about are Stargardt's disease and leukemia. Now, Stargardt's disease is a genetic disease which first presents itself in childhood. Hang on a minute. Okay, I was just looking it up. Cord blood cells can differentiate into some body cell types, not all body cell types. FYI, I'm just correcting myself. Okay, Stargardt's disease is genetic and it first presents in children, and it leads to a gradual loss of vision due to malfunction in retinal cells. Now, in order to treat Stargardt's disease, you can inject embryonic stem cells into the eye. If they're not rejected, they can attach to the retina and help to improve the vision. The other disease you need to know about the treatment of is leukemia. So it's a type of cancer of the blood, which is characterized by an increase in the number of white blood cells, and it's caused by a mutation. This can be treated, but what you would do is you remove healthy adult stem cells from the bone marrow. You would then treat the leukemia with a cocktail of chemotherapy. Once all of the cancer cells are dead, you can then put the healthy adult stem cells back in to the individual, because they will then make new blood cells. Now, the difference between Stargardt's and leukemia is that in leukemia, you're using the patient's own cells in order to treat the disease, so therefore they're definitely not going to be rejected, whereas in Stargardt's disease, they're just using generic embryonic stem cells, so there is a chance of rejection. Now, the use of stem cells, it has a number of ethical issues. For embryonic stem cells, the ethical issues include the fact that the embryo has to be killed, and as I just said, they're genetically different to the recipient cells, so they might reject them, and there's also a risk of them becoming tumour cells once they've been injected. With cord blood cells, they are limited in their quantity because they're only in the umbilical cord. However, the umbilical cord is always discarded, so if you don't take the stem cells, then they're going to be discarded anyways. And adult stem cells are difficult to obtain because you have to go through a procedure in order to get them, and they can only differentiate into a few cell types because they're multipotent. So they have limited differentiation capacity. They can't turn into anything, only a few cell types.